diffuse large B-cell lymphoma, we've recognized over the past several years is not a single entity, but is comprised of multiple subtypes, originally described as germinal center and activated B-cell subtype. And more recent advanced molecular profiling has demonstrated that within each of these groups, there are high risk and low risk subsets. And that risk stratification is described with frontline chemotherapy treatments, but it hasn't really been explored in the setting of chimeric antigen receptor T-cell therapy. So the purpose of the study was to determine the impact of these subtypes on the outcomes of patients treated with CAR T-cell treatment. Right, so we sought to collect biopsy specimens from patients with relapsed or refractory diffuse large B-cell lymphoma, all of whom were treated with commercial CAR T-cell therapy across 13 U.S. academic medical centers. We started with over 100 cases, and after applying genomic analysis, including gene expression and next generation sequencing to these cases, the final interpretation was applied to 96 biopsies. The subtypes, according to the two dominant modes of subclassification, was assigned, and then the treatment outcomes were described. What we discovered is that actually the patients who are traditionally thought of as being the highest risk, or some of the highest risk groups, including those with so-called cluster 5 or MCD subtype, as well as those with what's called the A53 subtype, which has preponderance of P53 mutations. These are so-called cluster 2 patients. All of these patients in these groups had very similar outcomes to the other lower risk groups. The only signal that we saw was for a trend towards inferior progression-free survival in the so-called cluster 3 patients with EZB subtype. And this suggests that CAR T-cell therapy can overcome some of the high-risk features that we see in the frontline setting. Well, we know that patients with relapsed refractory large B-cell lymphoma still have a certain degree of failure with CAR T-cell therapy. Probably about 40% of patients achieve durable remission, which means that up to 50 or 60% have failure to respond durably. We did look at some of the mutations in these biopsies that were associated with increased risk of treatment failure. And we did find that likely activating mutations in MYC and BCL2 were associated with treatment failure, as well as a few other mutations, including some cyclin-dependent kinase inhibitor 2A and KLHL6. So I think we'll need more data to confirm these in larger prospective series or validation cohorts. But as we seek to define the molecular features that may confer a poor outcome with CAR T-cell therapy, there may be strategies going forward which would allow us to intervene perhaps earlier or at the same time as CAR T-cell treatment to improve the outcomes of these patients with high-risk features.